Think about how much we rely on pathologists looking at tissue samples under a microscope. I mean, it's really a cornerstone of medical diagnosis, isn't it?
Absolutely. It's fundamental.
Now, imagine those microscopic slides becoming digital images. Suddenly, computers can get involved in analyzing these cellular landscapes, and that's precisely what's happening with the rise of whole slide imaging.
Yeah. And that brings us right to the fascinating topic of this deep dive. Artificial intelligence in histopathology image analysis. We've gathered some excellent sources that really map out this rapidly advancing field. We're talking everything from, established computational techniques right up to the very latest breakthroughs in deep learning.
So our goal here really is to kind of make sense of it all for you. How is AI actually being used to interpret these digitized tissue samples at, well, a microscopic level?
Right.
We want to give you a clear understanding of the key developments in this pretty complex area.
It's definitely a critical area, and it's worth remembering that analyzing these images is far from simple. I mean, there's a huge amount of visual information just stemming from the inherent complexity of biological tissues. But it's also affected by things like variations in staining techniques, the orientation of the tissue structures, even the magnification used when the slides are scanned. It all adds layers of difficulty.
Right. A cell isn't going to look identical in every single image. That makes sense. And from a computer's perspective, you can imagine it gets even more complicated with issues like cells overlapping, maybe, or the boundaries between different tissue types not being sharply defined.
Exactly. Getting a computer to accurately see these images involves tackling some real challenges. Things like distinguishing between similar-looking structures, dealing with partially hidden objects, maybe inconsistent lighting across the image.
Yeah.
It's tough.
Okay, so facing all those complexities, where did researchers even start? I mean, trying to get computers to analyze these visual details.
Well, before the deep learning revolution really took hold, there were essentially two main paths, what we generally call hand-designed methods and trainable methods.
Hand-designed. Okay, that sounds like someone was meticulously programming in very specific rules for the computer to follow, maybe?
That's a pretty good way to picture it, actually. Hand-designed methods work by looking for patterns based on fundamental pixel characteristics, things like their color, their texture, their shape. The idea was to use these characteristics to, well, break down the image into meaningful regions.
Okay. What were some of the specific techniques they used in that approach?
One set of popular techniques were active contour models. You can think of these as flexible lines or shapes that start somewhere in the image and then dynamically adjust themselves to try and latch onto the boundaries of objects.
Oh.
You had approaches that focused on edges, like geodesic active contours or GAC, and others that looked more at regions, such as the Chanves method, CV. For instance, the Chanves technique was applied quite a bit to segment glands in images of prostate cancer.
That sounds like a useful tool. Were there limitations? What were some of the downsides?
Oh, definitely. They could be quite sensitive to where you initially place that contour, the starting point.
Right.
Also, if you had multiple separate objects close together, it could be difficult for the model to handle them individually. They might merge or get confused.
Interesting. Were there other kinds of hand-designed methods being explored?
Yes. There was a method using something called a fuzzy sign pressure force function that proved quite effective at segmenting epithelial regions in colorectal cancer, even when the images had a fair amount of noise.
Huh.
Researchers also explored combining different techniques. So, for example, in analyzing prostate cancer, they combined color gradient geodesic active contours with hierarchical normalized cuts. The aim there was really to make the process more robust and, ideally, require less manual setup or user intervention.
So they were trying to leverage the strength of different approaches, kind of mix and match.
Exactly. And a key insight that emerged from this work, I think, was the value of focusing on morphometric and shape features in the analysis. So describing the objects based on their forms rather than just their colors.
Right.
By doing that, they could develop solutions that were less susceptible to those variations in how the tissues were stained in the lab.
That makes a lot of sense, focusing on the fundamental structure rather than something that can change easily. So, okay, that's the hand-designed era. What about these trainable methods you mentioned?
Right. So these methods, also known sometimes as classical or statistical machine learning, introduce the concept of a distinct training phase where you show the computer examples and then a testing phase where it applies what it has learned to new unseen data.
Okay.
It's a bit like teaching a student using practice problems and then giving them an exam.
Got it. How did this training process actually work back then?
The training, which usually happened offline, typically involved a few key steps. First, the images were pre-processed, normalization, maybe isolating the regions of interest. Then came feature extraction, where various characteristics were calculated from these regions, texture, shape, color intensity, things like that.
Mhm.
Finally, this extracted data was used to build a model, either a classifier to assign labels, like cancer or not cancer, or perhaps a probabilistic model to understand the underlying patterns.
And then the testing phase put that trained model to work on new stuff.
Exactly. In the online testing phase, they would take new, unseen images, perform the same pre-processing and feature extraction steps, and then use the trained model to predict the type of each pixel or region in that new image.
So they were essentially teaching the computer to recognize specific visual signatures based on the features they had defined beforehand. What kind of trainable methods were common?
We saw the use of Bayesian classifiers quite a bit. They looked at color, texture, and morphology, shape basically, to help determine the grade of prostate and breast cancer, and even sometimes to distinguish between cancerous and non-cancerous breast tissue. There was also Bayesian voting used to identify and remove stromal cells based on their texture and color features. And interestingly, fuzzy C-means clustering was even used sometimes to help initialize those active contour models we talked about earlier, specifically for segmenting cell nuclei.
It sounds like there was a lot of creative combination going on, using different computational tools together.
Absolutely.
Mm.
And there are also what we call discriminative approaches, things like k-nearest neighbor, self-organizing maps or SOMs, random forests, and support vector machines, SVMs.
SVMs.
Think of SVMs as trying to draw the clearest possible line or boundary between different categories of cells in the image, even if those categories look quite similar. These methods really focused on directly learning the decision boundaries between different classes. For example, SVMs were used in a patch-based method to segment squamous epithelium in images of cervical tissue.
Patch-based. Did that introduce any particular challenges using patches of the image?
It did, actually. That particular method turned out to be quite computationally intensive. It took a lot of processing power, and sometimes it led to oversegmentation, where it would break up regions that probably should have been considered as a single whole unit.
Oh, okay.
And that kind of points to a common limitation of many of these pre-deep learning methods, both hand-designed and viewable. They often resulted in segmented objects with gaps or poorly defined edges. Frequently, they required a whole separate cleanup step afterwards, some kind of post-processing.
Okay, so that gives us a good picture of where things stood before the big shift. Then came deep learning, which has really transformed so many areas of AI. How did it change the landscape of histopathology image analysis?
Oh, profoundly. Deep learning, and specifically convolutional neural networks, or CNNs, has achieved just remarkable success in both segmenting and classifying structures in histopathology images.
Yeah.
The major advantage here is that CNNs can automatically learn hierarchical features. You can almost imagine it like this. The early layers learn to recognize basic visual elements like edges and corners, simple stuff.
Right.
And then the deeper layers combine these basic elements to identify more complex structures, like different types of cells or tissues. It builds up complexity.
So instead of researchers having to manually define features like color and texture beforehand, the network kind of figures out what the important features are on its own directly from the image data. Is that the idea?
Precisely. That's the core difference. And this ability to automatically discover relevant features has led to really significant advances. Maybe let's start by looking at how deep learning is used for tissue classification.
Okay, yeah. What kind of progress have they made in classifying different types of tissues or cells using these deep learning approaches?
Well, we've seen the development of multi-stage CNNs, for instance, like a two-stage CNN designed specifically for classifying nuclei in breast cancer images. Researchers have also explored CNN ensembles quite a bit. This involves combining the predictions of multiple CNNs, often with different designs or architectures, to improve overall accuracy.
Right.
The key insight there is that by combining different architectural strength, these wisdom of the crowd approaches can sometimes catch errors individual networks might miss. This leads to more robust and accurate diagnoses, especially when you're dealing with the inherent complexity of tissue samples.
Mm-hmm.
Examples include things like EMSNet and hybrid CNN plus LSTM architectures, and also ensembles of well-known networks like VGG19, MobileNet, DenseNet, ResNet, Inception v3, and Inception ResNet v2. Lots of combinations being tried.
Why go to the effort of combining so many different networks? Is it just about boosting accuracy, or are there other reasons?
Boosting accuracy is a big part of it, definitely, but ensembling can also be particularly helpful when dealing with those very large whole slide images. You often break them down into smaller patches for analysis, and combining results from different network perspectives on those patches can be beneficial. Techniques like feature concatenation, where you literally combine the learned features from different networks, and transfer learning have also been really effective.
Transfer learning. That's taking a network trained on one thing and applying it somewhere else.
Exactly.
Yeah.
You take knowledge learned by a network on a massive data set like ImageNet, which has millions of everyday images, and you apply that foundational knowledge to the more specific domain of histopathology images. It often gives the network a good head start.
And what happens when you don't have huge amounts of labeled data to train these complex deep learning models? That seems like it would be a common challenge in medical imaging, getting all that detailed labeling done.
Oh, it's a critical issue, a huge bottleneck. So researchers have investigated methods like unsupervised pre-training followed by supervised fine-tuning.
How does that work?
Well, in this approach, the network first learns general visual features from a large amount of unlabeled data, just looking at patterns in the images themselves. Then it's fine-tuned on the smaller, available labeled dataset for the specific classification task you care about.
Okay, so it learns general stuff first, then specializes.
Exactly. And generative adversarial networks, or GANs, you might have heard of these things like histogram, have also been used for synthetic data augmentation.
Creating fake images, essentially.
Sort of, yeah. Essentially creating artificial but realistic-looking images to increase the size of the training set. You can think of GANs as having a counterfeiter network, the generator trying to fool a detective network, the discriminator, into believing its fake images are real.
Right.
This adversarial process pushes the generator to create increasingly realistic synthetic data, which can then supplement the real labeled data.
That's a clever way to work around the limited data problem.
Indeed. And for analyzing whole slide images where you might only have a label for the entire slide, like this slide contains cancer, but not for individual cells or regions, techniques like multiple instance learning or MIL have been developed, and variations like correlated MIL, such as transbellum. These methods can make predictions at the slide level even when only these coarse image level labels are available. There are also frameworks using things like unit stochastic selection and attention fusion specifically for WSI diagnosis, as well as hybrid supervised learning approaches that try to combine information from both image level and pixel level annotations if you happen to have both.
It really sounds like they're attacking this data scarcity problem from multiple angles.
They absolutely are. Cluster to conquer, or C2C, is another interesting method for WSI analysis. It divides the image into smaller patches, groups similar patches together into clusters, and then uses attention mechanisms to focus on the most informative groups for making the final diagnosis.
Focusing on the important bits.
Right. And when you have some labeled data, but also maybe data from different sources, different hospitals, different scanners with slightly different characteristics, deep transferable semi-supervised domain adaptation models like HisNet SSDA have been developed. These try to transfer knowledge from a more heavily labeled source dataset to a partially labeled target dataset, adapting to the differences between them.
Okay, so that covers classification pretty well. What about using deep learning for tissue-level segmentation? So trying to actually delineate different tissue types or regions within the image.
Yeah, that's another major area. We've seen the development of networks like the Deep Contour Aware Network, or DCAN. It uses a technique called multitask learning to simultaneously perform the segmentation and identify the boundaries, the contours of those segments.
Doing both at once.
Exactly.
Oh.
And that helps in situations where the visual differences between tissues might be subtle or the boundaries aren't very clear-cut.
Now, I've been hearing a lot about transformers in the broader AI field lately. Are they making an impact in histopathology image analysis as well?
Absolutely. Vision transformers, which use these self-attention mechanisms to understand the relationships between different parts of an image, even distant parts, are being explored quite actively. They're looking to address some of the limitations of traditional CNNs in capturing long-range dependencies or global context. And we're also seeing hybrid models that combine the strengths of both worlds, so using CNNs for their power in local feature extraction and transformers for understanding the bigger picture, the global context. A good example is TransUNet. These hybrids are showing really promising results in improving segmentation accuracy.
But is there a downside?
Well, it's often a trade-off. These more complex models, especially transformers and hybrids, can sometimes require significantly more computational resources and longer training times compared to standard CNNs.
Right, so potentially better performance, but at a higher cost, computationally speaking. What are some specific, maybe real-world applications of deep learning in this kind of tissue-level segmentation?
One area is segmenting epithelium tissue. There's a model called PISO, for example, which is a deep learning algorithm designed specifically for segmenting epithelial tissue in prostatectomy slides. It actually uses a very precise staining technique called immunohistochemistry as a kind of gold standard for comparison during training and evaluation. CNNs and also recurrent neural networks, or RNNs, have also been used to classify different types of epithelial tumors in the stomach and colon based on analyzing whole slide images.
And what about drilling down even further to the cellular level, trying to segment individual cell nuclei? That seems incredibly challenging.
It is challenging, but there's been significant progress there as well. ASPPUNet, for instance, uses a technique to detect concave points on the apparent boundary of cell clusters to better separate nuclei that are overlapping.
Dealing with overlap.
Exactly. NucleSegment incorporates things like residual connections and attention mechanisms to improve nuclei segmentation, specifically shown in liver cancer images. CIANet focuses on aggregating information in a way that's specifically aware of the contours of the nuclei to try and improve accuracy.
Lots of specialized networks.
Definitely. We've also seen approaches that combine simpler segmentation methods like superpixels with the learning power of CNNs, and methods like multi-scale adaptive nuclei analysis, or MNA, which uses adaptive thresholds based on the characteristics of individual objects rather than a single threshold for the whole image.
It sounds like a real focus on getting down to that fundamental unit of biology, the individual cell, and doing it accurately.
It really is. And then you have even more advanced methods like HoverNet, which can simultaneously segment and classify nuclei by estimating how far away each pixel is from the center of a nucleus using these distance maps.
Wow.
Mesmer is another advanced pipeline that uses components called PanopticNet and TissueNet to perform both nuclear and even whole cell segmentation. And because histological structures often have these really irregular, complex boundaries, those transformer-based models we mentioned, with their ability to understand both local details and global context, are proving particularly useful for getting accurate segmentation in these tricky cases.
This is all incredibly detailed work. Now, I know one of the major goals, kind of the holy grail for AI in medicine, is to advance precision oncology. How is deep learning in histopathology contributing to that bigger picture?
That's a great question, and it's a really exciting area. Histopathological features the patterns pathologists see under the microscope often provide crucial clues about the underlying molecular characteristics of a tumor and how a patient's disease might behave clinically.
Okay.
So deep learning is now being used to try and predict genetic alterations directly from these tissue images.
Just from the image.
Just from the standard H&E stained image in many cases. There's also growing research showing that we can directly predict clinical outcomes, things like patient survival time, directly from the visual patterns in these images across a whole range of cancer types.
That's remarkable. I mean, the idea that the visual patterns we've been observing for decades can actually tell us about the hidden genetic changes and the likely course of the disease, that's powerful.
It really is. Models are being developed, for instance, to predict things like whether colorectal cancer is likely to have spread to the lymph nodes just based on the primary tumor image. We're also seeing multitask deep learning approaches. These are models trained to predict multiple things at once, say, both patient outcome and the status of specific biomarkers, like hormone receptor status in breast cancer. These are being used for improving breast cancer prognosis.
Right.
The potential here, the big promise, is that these detailed computational analyses can extract much more information, more subtle patterns from the images than traditional human visual assessment alone might capture.
But I imagine that requires a huge amount of data linking the images to the detailed clinical and molecular information, right? You need that ground truth.
Absolutely. That is the key requirement. Large, well-annotated data sets that include comprehensive treatment information, patient outcomes, genetic data that's essential for training these kinds of predictive models effectively.
Makes sense.
And in precision oncology, it's often important to analyze images from different staining techniques or different biomarkers together for the same patient. So image registration models become important.
Aligning the different images.
Exactly, aligning these different views of the same tissue. There are intensity-based methods that look at overall pixel values and feature-based methods that focus on specific landmarks. While dealing with the variations in staining between different techniques remains a challenge, those feature-based methods tend to be a bit more robust for this task.
Okay, we've talked a lot about the power of deep learning, but a recurring theme has definitely been the need for large amounts of labeled data. You mentioned it's a bottleneck. How are researchers tackling this problem of scarce annotations head-on?
Yeah, this is a very, very active area of research because it's such a practical problem. Two particularly promising approaches that have gained a lot of traction are weakly supervised learning and self-supervised learning.
Okay, let's start with weakly supervised learning. What's the core idea behind that? How does that help?
Weakly supervised learning aims to train models using less detailed or weaker forms of annotation. So, for example, instead of having pixel-perfect labels outlining every single cell type, you might only have a label for the entire image or the whole slide indicating whether it contains cancer or not. Just a single label for a huge image.
Right. Much less work to create that label.
Much less work. And we're seeing really interesting things like privacy-preserving federated learning approaches. These use weakly supervised multiple instance learning, that MEL technique, again on massive data sets of gigapixel images pooled from multiple hospitals, often using just those slide-level labels.
So you can learn from less precise labels, but potentially across a much larger and more diverse collection of images from different places while preserving privacy.
Precisely. That's the idea. There are also interpretable weakly supervised frameworks being developed that can predict patient survival using whole slide images, and they only need the overall survival time of the patient as the supervisory signal, as the label. Methods have also been developed for classifying and even roughly locating carcinomas, finding where the cancer is in the lungs and colon, using only these global image-level labels. No detailed annotation needed.
Okay.
Dual stream networks are being explored to combine information from small image patches, the local view, with the overall context of the whole slide image, the global view. And hybrid CNN-based methods are even incorporating data from other modalities, like MRI scans, using weakly supervised patch selection to help classify different types of gliomas, a type of brain tumor.
It really sounds like they're trying to squeeze every last drop of information out of these less granular labels.
Exactly right. HCRF-AM and IL-MSAM are just further examples of frameworks that use attention mechanisms and sometimes interactive learning to perform weakly supervised classification of histopathology images.
Is there a catch?
Well, the main drawback often is that weakly supervised methods can be more complex to design and train, and sometimes they're more computationally intensive than fully supervised approaches because the model has to infer more from less direct guidance.
Okay, that makes sense. And then the other approach you mentioned was self-supervised learning. How does that differ from weakly supervised?
Self-supervised learning, or SSL, makes a really different approach altogether. Instead of relying on any external labels, even weak ones, it learns meaningful features directly from the raw, unlabeled image data itself.
How does it do that without labels?
The core idea is to create artificial pretext tasks where the network has to learn something inherent about the structure of the data. For example, predicting a missing patch in an image, or whether two different augmented views came from the same original image. By solving these self-generated puzzles, the network learns powerful visual representations, powerful features that capture the underlying structure of histology images. And this learned knowledge can then be transferred very effectively to downstream tasks, like classification or segmentation, even when you only have a small amount of labeled data for that specific task.
That sounds almost like the AI is teaching itself to understand the visual world of histology before tackling a specific problem.
That's a great way to put it. In a way, yes. For example, there's a multiple instance learning technique that uses a new way of aggregating information combined with self-supervised diverse learning to categorize whole slide images and identify tumor regions without needing any precise annotations of the tumor boundaries at all.
Impressive.
Transpath is another example. It's a hybrid CNN transformer model that is first pre-trained on a large collection of unlabeled histopathology images using self-supervised learning. It learns the general visual characteristics of tissue first.
Okay.
Graph convolutional networks, or GCNs, combined with self-supervised pre-training are also being used. These can incorporate contextual information not just from the tumor itself, but also from the surrounding healthy tissue, learning relationships between different regions.
So by learning these general patterns from unlabeled images, these models become much better starting points for a variety of specific tasks, even if you only have a small amount of labeled data for those tasks.
That's exactly the core idea and the big benefit. We're seeing task-agnostic, unsupervised representation learning that leverages information from the image at multiple resolutions and in different contextual regions. Pre-training on vast amounts of multi-organ datasets using a technique called contrastive self-supervised learning is proving really effective in improving the quality of the features the network learns.
Contrastive learning. What's that involve?
Contrastive learning generally involves teaching the model to pull representations of similar images, like different augmented views of the same patch, closer together in feature space, while pushing representations of dissimilar images further apart.
Got it. Like learning what makes images similar or different.
Exactly. And it's powerful. Deep Smile, for instance, uses contrastive self-supervised pre-training to predict important molecular markers like HRD and MSI status. Contrastive predictive coding, another SSL method, has shown great promise in developing feature extractors that achieve excellent performance in tasks like MSI prediction, even when only a very few labeled examples are available.
It really sounds like self-supervised learning could be a genuine game changer for overcoming that annotation bottleneck in this field.
It absolutely has tremendous potential. Researchers have even found that using specialized neural network backbones, the core architecture of the network, that are pre-trained specifically on H&E stained images using self-supervised methods like BLL, can actually outperform those general-purpose backbones pre-trained on broad data sets like ImageNet. Specificity helps.
Interesting.
And self-supervision signals, based on something as simple as identifying the magnification level an image was taken at, have even been used to help pinpoint the locations of nuclei. So the future of the field may very well involve more generalizable techniques trained on massive amounts of unlabeled data, significantly reducing the burden of expert annotation for highly specialized tasks, especially with the ongoing advancements in techniques like contrastive learning.
All this talk about AI making complex predictions, especially in medicine, raises a really important question. How do we know if we can trust these predictions in a clinical setting? How do we peek inside the black box? That brings us to the crucial area of transparency and understandability, or what's often called explainable AI, XAI.
Exactly. That's absolutely critical. For AI to be truly valuable and safely integrated into real-world clinical workflows, we need to understand not just what the AI is predicting, but also why it's making that prediction. Uncertainty quantification is often considered an important initial step towards achieving explainability. It basically involves trying to measure how confident the model is in its own predictions.
Okay, so not just the answer, but how sure it is of the answer. How do they go about measuring this uncertainty?
Well, one way is by calculating something called Shannon entropy on the probability distribution the model outputs for its prediction. A low entropy score generally indicates that the model is quite certain, concentrating its probability on one outcome. Another very common approach uses a technique called Monte Carlo dropout, or MC dropout. This is a way of getting multiple slightly different predictions from the same model on the same input image by randomly dropping out some neurons during prediction.
Okay.
By looking at the spread or variability, like the standard deviation of these multiple predictions, we can get a quantitative sense of the model's uncertainty. A wider spread implies higher uncertainty.
So if the model gives slightly different answers each time you run the same image through it with this dropout enabled, that suggests it's not very sure of its initial prediction.
Precisely. That's the intuition. And recent research has started incorporating these measures of uncertainty directly into their models, sometimes achieving better segmentation and classification results by leveraging this uncertainty information. They've also used uncertainty to create what are called instability maps, which visually highlight the regions in an image where the model is least confident in its analysis.
Okay, that's useful. And then we have the explainability techniques themselves, the XAI methods. How do these help us understand why a model arrived at a particular prediction? What does it look like?
Right. So these techniques aim to provide insights that can be meaningful potentially to both the AI developers trying to improve the model and, importantly, to the clinical staff, the pathologists, the doctors who would actually be using these tools. For clinicians, visualizations directly on the input images are often the most helpful and intuitive. Heat maps are very common. You might have seen these. Methods like GradCam overlay a color gradient onto the original image, where maybe warmer colors indicate the pixels or regions that were most influential in the model's final prediction.
Right.
Patch selection methods work differently. They systematically hide or block different parts of the image and observe how the network's output changes. This helps reveal which regions are most critical for the decision. Examining the receptive fields of the neurons in the network can also help understand which areas of the input image are being looked at by different parts of the model when it makes a decision.
So a pathologist could potentially look at one of these heat maps and see, the AI flagged these specific cells or this particular region as being most important for its diagnosis.
Exactly. That's the goal, to provide that kind of visual grounding. Attention-based methods, like those used in a network called SUNet, also provide a form of visual explanation by inherently highlighting the regions in the image that the network is paying the most attention to when processing it. Then there are synthetic visualizations. These are a bit different. Methods like guided backpropagation, deconvnet, integrated gradients, smooth grad. These are more about assessing how sensitive the model is to different kinds of input patterns or features.
More for the developers.
Generally, yes. While perhaps less directly used by clinicians in their day-to-day practice, these are really valuable tools for AI developers and for quality assurance. They help understand how the model might respond to different variations in image data, potential biases, things like that.
That makes sense. The
Yeah.
developers definitely need to understand the inner workings too. Are there other ways to explain a model's behavior beyond these kinds of visual techniques?
Yes, definitely. Clustering explanations are another approach. These involve grouping input images together based on how the neural network processes them internally, perhaps based on their feature representations in some layer.
Okay.
By visualizing these clusters, for example, using a dimensionality reduction technique like TSNE embedding, you can start to see what characteristics the network is using to group similar cases together. It can reveal the model's internal rationale and even help identify unusual or outlier cases that the model handles differently. Another really interesting approach involves using actual examples from the original training dataset to explain a prediction. For instance, one study used those generative adversarial networks, GNs, to transform patches of cancerous tissue to look visually similar to healthy tissue.
Like a visual counterfactual.
Exactly. The idea is that showing pathologists this kind of visual transformation, here's what the AI thinks needs to change for this to look healthy, could help them build more trust in the model's ability to correctly identify the cancerous regions in the first place.
It's almost like providing a visual before and after scenario based on the AI's understanding to build confidence.
Precisely. And it's worth mentioning there are also several open source frameworks and software tools becoming available now. Things like ExplainX, Google's explainable AI framework, libraries like InterpretML, Skater, AI Fairness 360. Lots of resources providing a wide range of methods for understanding and explaining AI models.
Okay, so we're getting reliable predictions, hopefully, and we're working on making them interpretable. But the survey materials also touched on this idea of actionability. What does that concept mean in the context of AI-driven histopathology? It sounds important.
It is really important, and it's kind of the next logical step. Actionability goes beyond just providing a prediction and maybe an explanation. It's really about the ability to translate those insights into concrete actions that can directly benefit patient care.
Like what?
Like suggesting a specific treatment based on the predicted subtype of cancer, or recommending further diagnostic tests, maybe suggesting a specific follow-up scan. Ultimately, the goal is for these AI tools to not just inform clinicians, but also to actively guide or support clinical decision-making in a useful way.
It makes perfect sense. I mean, a diagnosis or a prediction is only truly valuable if it leads to some kind of beneficial action for the patient, right?
Exactly. The survey describes a potential workflow where the predictions from a deep learning diagnosis model are fed into a separate actionability component. This component might use factors like the model's confidence in its prediction, that uncertainty measure we talked about, to decide on the next step. So, for example, if the AI is very certain about its diagnosis, it might automatically generate a preliminary report or suggest a standard treatment pathway. However, if the AI is less certain, it could flag the case and explicitly recommend review by a human pathologist, perhaps highlighting the uncertain regions.
So using the AI's own assessment of its uncertainty to determine the appropriate level of human involvement or the next clinical step.
Exactly. That's one potential way it could work. However, the survey does point out that developing these kinds of truly actionable recommendations that are safe, effective, and integrate well into complex clinical workflows is currently quite challenging and still in the relatively early stages of research and development.
It definitely sounds like that's a critical area where future research and development really need to focus, bridging that gap between prediction and action. Finally, how do we actually evaluate whether these explanations that AI provides are any good? How do we know if they are genuinely helping the users, the pathologists, the clinicians, understand and trust the system appropriately?
Yeah, evaluating explainability itself is a challenging but incredibly important area. It's not straightforward. There are human-centered evaluation methods that try to look at how well the explanations help users develop an accurate mental model of how the AI actually works.
Okay.
Studies often use things like questionnaires and Likert rating scales to measure how much users feel they understand the AI's reasoning and how much they trust the explanations provided.
So directly asking people about their subjective experience with the explanations.
Yes, that's a big part of it. Experiments also try to assess more objectively whether users can better predict the AI's output on new cases when they're provided with explanations compared to when they are not.
Testing their understanding.
Right. And these studies have shown that good explanations can indeed improve a user's understanding of the model's behavior and, importantly, reduce the tendency for users to overestimate the AI's accuracy, which is a risk. However, these kinds of qualitative assessments, relying on user feedback and observation, can be subjective, sometimes biased, and often quite time-consuming to conduct properly.
Sure.
So researchers also look at more objective measures. Things like how quickly users can make decisions or complete specific diagnostic tasks when using the AI tool with explanations versus without. They might also look at subjective ratings of the difficulty of those tasks.
Okay.
Trust and reliance are also key factors. Researchers might measure how often users agree with the AI's recommendations and how that agreement level changes when explanations are provided versus when they are absent. Does seeing the why change their willingness to follow the AI's lead?
Right.
A more general evaluation method is sometimes just to measure the time it takes users to perform a task before and after the introduction of explanations, looking for efficiency gains or losses.
It sounds like a combination of trying to measure understanding, trust, efficiency, and reliance using both subjective and objective methods.
Exactly. It's multifaceted. And importantly, there are also computational methods being developed to evaluate the explanations themselves, independent of human users initially. The goal here is to move beyond just creating explanations that are merely convincing to humans, which might mask underlying model flaws. Instead, the aim is to develop methods that assess whether the explanations are truly accurate, faithful, and transparent descriptions of how the model is actually functioning internally.
This has been a truly comprehensive exploration, really, into the world of AI and histopathology image analysis. We've journeyed from those early days of hand-designed features and classical machine learning all the way to the sophisticated deep learning models of today.
Yeah, models capable of incredible feats like detailed tissue classification, intricate cell segmentation, and even making predictions that are directly relevant to precision oncology. It's been quite a leap.
And we've also highlighted the significant efforts being made to overcome those real-world limitations, particularly the need for vast amounts of labeled data, through innovative approaches like weakly supervised and self-supervised learning.
Absolutely. And crucially, we spent time discussing the vital role of explainability, the XAI piece, and also actionability. These are essential for ensuring that these powerful AI tools can eventually be effectively, ethically, and safely integrated into real-world clinical practice, building the necessary trust and enabling practical applications that actually benefit patients.
So as AI becomes an increasingly integral part, it seems, of pathology workflows, it really prompts us to think, how will this collaboration between the, irreplaceable expertise of human pathologists and the ever-growing computational power of AI continue to shape the future of diagnostics? It feels like we're right on the cusp of something significant.
I think we are. And what novel insights into the complexities of disease will this powerful synergy ultimately reveal? It really does raise profound questions, not just technologically, but also about the ethical considerations and the very trajectory of diagnostic medicine going forward.
A field just brimming with potential, it sounds like. And the advancements we've explored today are, well, undoubtedly just the beginning of what looks to be a truly transformative era.
Couldn't agree more. Exciting times ahead.